Next talk is by Roberto Caminiti from Sapienza University in Rome. His talk is entitled On the Local Nature of Command Functions in the Parietal Frontal System.
It has been a very long day. Yes, but I don't see the image there. I'm saying that it has been a very long day. We are all very tired, and I would like to start from the end of my talk, and if I will be able to start from the end of my talk, I would like to flip things over. So I'll ask question first to you, and the first question, after saying, of course, thank you very much for inviting me, Oksana and the organizing committee, the first question is to Nikos. So where are you, Nikos, here? You are probably at the wrong meeting. We are at the wrong meeting, is that right? So do you remember where are you first? You are, this is you? Okay. And so, it's Strasbourg, so you don't need your engram cell to be reactivated by Dr. Tonegawa. There are other people here. Miki, you are busy with your phone, but where are you? You are here, okay. Well, this is a meeting that I did organize with Klaus-Peter Hoffmann. Where is Klaus? Klaus? Here is Klaus. Okay. Okay, good. So, good memories. About 26 years ago. Now I have another question for you, Nikos. Where are you here? Well, you always say that Italy is your second country and you love Italian culture. Please tell me, where are you here in this picture? No. Well, I would say that this is you, is St. Thomas, because doubt is what promotes science, is the first mover of science. So thanks for all the science you have done so far for us and for everybody. And thanks for putting this together, which is possible thanks to you. Now, I refrain myself to ask, well, I don't want to be tempted to ask Miki and Klaus-Peter, where are you in this picture? Because we might get very embarrassing responses. Okay. Okay, so if I was successful in raising your attention, it's very late in the afternoon, I'm going to talk about, basically, about motor cognition and neural circuit for motor cognition, which in classical terms is the what's happening in the dorsal stream of our cerebral cortex. But let me define what are the precursor of these ideas, just to put things in some sort of general frame. So I believe that the field started many years ago with the intuition of a very intelligent and influential Swiss psychologist, which is Jean Piaget. In his studies of child development, he made it clear that the absolute cognitive abilities of adults are constructed on the basis of the sensory motor interaction that we experience as a child. And this idea was somehow mutuated into the experimental psychology on animal with a famous experiment by Held and Heine in 1963, who have shown that the visual behavior of newborn kitten does not develop properly if they are not allowed to exert their own active control on their visual experience. You see two kittens, one is allowed to move around, the another one is in a gondola. He can look around, he has normal vision, but this goes over and over for days and days. And then when this kitten is free, he doesn't know what really to do. And his main problem is a problem of distance perception and encoding. So that the hit obstacle is uncertain during locomotion and things like this. These apply also to motor behavior on monkeys where they are not allowed since their birth to reach a visual target in the extrapersonal space. When their arm is free, is clumsy, it's very hesitating, and when the monkey finally learn how to reach at an object of interest, at a raisin or something like that, the other limb, which has not been experienced the same thing, doesn't know almost anything, because in absence of evacuative movement, even interhemispheric transfer of manual information is prevented. I mean, within this general field, I believe that one of the most influential view, which has entirely shaped, I would say, the field of motor cognition, is the ecological view of behavior by Gibson. His main point was perception is not just passive construction of of an internal representation of the external world, but is searching in an active fashion for information of interest, for salient information in the extrapersonal space. And this is related to the concept of affordance, which is a very influential concept, which means affordance are opportunities for actions around us. And we'll see during my presentation what these might mean. Finally, the idea of the command function of the parietal lobe in a very influential paper by Mountcastle in 1975, where he for the first time presented a radically different view of the parietal cortex, not as an higher order sensory area where sensing is transformed into perceiving, but as a region of the brain which can command our operation, our hand and eye operation in the extrapersonal space. That the parietal lobe is somehow important for this kind of things. It's also nicely shown by this image, which I like very much. Paleoneurology tell us that if we compare the endocast of Neanderthals with the endocast of Homo sapiens, the difference is a marked globularization of the brain of the endocast over the parietal lobe. And this is accompanied by an increasing in length of the line which connect the parietal cortex to the cerebellum. All that region of the brain undergoes to an important expansion, which is probably related to the development of material culture, to the use of tools over the years. And this globularization is also evident in humans as compared to other non-human primates like macaque and marmoset, and is shown here. And this is also accompanied by the displacement of certain area. Here you have primary auditory cortex and area MT, and this is in monkeys, in macaque monkeys, this is where they end up in humans. Now, it is widely believed, based on neuropsychological studies in humans and the information that we have got in the last, let's say, 30 years from neuroanatomy and behavioral neurophysiology, that parietal cortex is parietal cortex and frontal cortex, and their relationship are the essential node for our action in the extrapersonal space. And this is a view of parietal cortex which shows, and of frontal cortex, which is based on a widely accepted parcellation, and this parcellation mostly come from connectivity studies. Parcellating parietal area based on architectonics is a very hard procedure. But some of these, in some of these areas, you can have reasonable and reliable parcellation based on architectonics, but most of it is done based on cortical connectivity, and basically there are 18 parietal areas. Some of them, 8 in the superior and 9 in the superior and 9 in the inferior parietal lobe, and some of them, you see them here, they are buried in the dorsal and ventral bank of the intraparietal sulcus, while when you go to frontal cortex, there is a much larger number of areas, including the orbitofrontal areas that account up to 36, 40 areas, which has been parcellated based on this connection. So the question is, it's nice that Katie has shown us the micro level of organization of connectivity at the level of single action. Now, here the task is, can we get a global view of all these connectivity? Well, there are different strategies you can use. And since neuroanatomy is a very purely quantified discipline, we try to inject some quantification, and we use some data-driven analysis, which is based on a full recollection of histological studies in monkey based on rate-target transport of tracer. This is a connectivity matrix only for the parietal areas, but we also built a connectivity matrix for the frontal areas, and then put all them together in a huge gigantic connectivity matrix, having all parietal and frontal area in rows and columns, and as a database where 100 means the maximum of connectivity, 67 is below, and so on up to 16. So we try also to quantify based on known, on declare by papers strength of connectivity, which is in any case a slippery ground, but is better than nothing. And so what you can do with this thing is just a canonical cluster analysis. And if you do a cluster analysis, you end up with image like these. These are clusters of parietal areas. This is the consensus tree, which tells you how many times each of these clusters is seen, occurs in the best 100 trees, which are restored by the cluster analysis. And you can see that in parietal cortex, you can have different clusters, which here are shown by same color. For instance, in the medial parietal part of parietal cortex, you have a cluster formed by area V6A, PGM or 7M, and area 31, which is this cluster here. In the dorsal bank of the intraparietal sulcus, you have a cluster which extends to the parieto-occipital junction and goes to the medial side of the hemisphere, which we call mediodorsal superior parietal cluster, then an anterior parietal cluster, and then a posterior cluster which include the area OPT, LIP, VIP, and then an anterior parietal cluster which include all the areas which lies in the exposed part of the inferior parietal lobe. Now, this cluster have in common connectivity. That's the take-home message. And I want to consider this cluster as an entry node for different task demand, because I do believe that clusters provide a richer functional repertoire to the request that task can impose, can ask to our brain than individual areas. And provided that they are all connected by local connectivity, otherwise they would not be clustering together, it is reasonable to think that this connectivity serves the purpose of deciding when a task imposes a question, which one of these areas can provide a better answer based on the congruence between the task demand and the functional properties of neurons within each of these. Now, the issue is how these things look in the frontal cortex. Well, in the frontal cortex, the picture is almost discouraging because but there is some logic in all of these. And the logic is that these are all the ventro-orbitofrontal clusters. It is a ventro-orbitofrontal cluster that combine all the areas which lies in the orbitofrontal cortex and on the lateral part of prefrontal cortex. They are almost secluded and they seem to stay on their own. Then you can have a dorsomedial prefrontal cluster, which is here, located on both the exposed and the lateral and the mesial part, and these include those which are considered the highest order areas in the monkey, like polar frontal cortex, the two subdivisions of area 9, which is considered the place of the theory of mind, whatever, for non-human primates, if they one. But there is some order, and then the posterior prefrontal cluster here. When you move to the frontal lobe, the singular area cluster together, and these all concern motor and premotor areas in the frontal lobe, with ventral premotor cortex areas like F4 and F5 and its subdivision clustering together, and motor cortex with the canonical or conventional premotor area, which are defined as the area which project directly to the motor cortex, again forming the so-called motor output system of the frontal lobe, because many of the corticospinal system of projection stem from there. But this is only part of the story I want to tell you. In this cluster, you see also areas like area F7, which include the supplementary fields, and area F6, which is the so-called pre-supplementary motor areas. They do not project directly to motor cortex. They do not project directly to the spinal cord, but they talk to premotor areas and to prefrontal areas. And they are considered a sort of interface between the executive control system in the frontal lobe, where strategies and tactics of behavior are supposedly conceived and represented, and the motor output system of the frontal lobe, because after all, even higher order executive function must find their way to the motor output. Now, the next step, which I will summarize because would be too tedious to go, is that this kind of clustering analysis give you also the connectivity between the different clusters. So you might know to which parietal cluster each individual frontal cluster is connected, okay? And if you do that, you can get an overall view, which here is not in form, is in a qualitative way, is not in form of arrows, which connect different brain regions, but in this system, colors, similar colors in parietal and frontal cortex connect clusters together. And when you connect clusters, which mean group of variables with similar functional properties, to a similar cluster in frontal cortex, you get what are the traditional information processing streams within the dorsal stream of the cerebral cortex. So this is the so-called dorsal reaching stream, is what we like to call dorsal reaching stream. This is which is very important for reaching, and it's interesting that lesion in this stream or reversible inactivation generate a syndrome which is very similar to optic ataxia in humans, and the corresponding region, when lesion in humans determine optic ataxia, there is another stream which is the lateral action, the lateral reaching stream, which is set in motion, supposedly, we believe, when we are engaged in action that lasts over a long period of time. So not for fast movement like reaching, but imagine object construction, tool use or something like this. And in fact, lesion in the corresponding part of the inferior parietal lobe in humans results in constructional apraxia or tool apraxia. So these patients have no motor impairment, no paralysis of nothing like this, no apparent movement disorder, but when they are called to copy an image or to construct a single object, they fail. Okay. Another stream that Miki would like is the one that include the posterior area of the ventral bank of the intraparietal sulcus, like area LIP, area VIP, some with premotor cortex, but some with the frontal eye fields. And this is what we can call an oculomotor intention and attention system. And finally, the so-called lateral grasping system, which has been studied intensively by people in Parma, by Hans Scherberger in Göttingen, which include the inferior parietal areas such as PF, AP, S2, and sectors of area F5 and area 12. Embedded within the system is the so-called mirror system. Now, this is a rather crude reconstruction of the processing stream, which we do believe emerged from the parietal frontal connectivity, but is only one part of the story. This is the same image here. And what you see here is exactly the same image. You just can imagine a flattening the brain and having this sort of couple. And this is just to present our conceptualization of this entire sector of the parietal frontal system. We like to think that the entire parietal frontal system is divided in at least 5 functional domains. A posterior parietal domain, an anterior parietal domain, a cingulate domain, a frontal domain, and a prefrontal one. the posterior parietal domain here is characterized by this high-end integrative domain at the parieto-occipital junction, which include area V6A, 7M, and OPT. And the reason we believe so is that this is the region where the visual input eventually coming from the occipital lobe, from the extrastriate area of the occipital lobe or from pulvinar, impinge on parietal cortex. And we believe that is a high-end integrative domain because if you record cell activity in behaving monkeys, in this case, this monkey had been trained in 7 different tasks where they were making movement in a center-out task in 8 different directions under normal reaching condition, reaching to a visual, to foveate and to foveate target, saccadic eye movement, memorize, reach to memorize target location, and so on. And when you plot all the preferred direction of every individual neuron, you find out images like these that we refer to as a global tuning field. So the neurons combine information about the retinal position of the visual stimulus, the position of the end, the movement, the direction of movement of the end, the position of the eye, the direction of movement of the eye, and also to memorize target location. Well, the glue of all these is directionality, which is a very pervasive signal throughout the entire parieto-frontal system. And each neuron has a global tuning field, and the size of the global tuning field is determined by the length of these orange vector, which is called mean resultant length. This is just directional statistics. But the overall distribution of the mean length of the global tuning field is uniform in space. So that it means that this parieto-occipital region contain an entire representation of the space for high-end action in a congruent fashion, and whenever we are called to move our eyes or our hand to a given part of space, the input will probably recruit those neurons which have functional properties, directional properties, which are more suitable for satisfying this task demand, which can be, and this can be done throughout the entire part of space. It's very important to remember that this is a uniform distribution, while when you go and do similar experiment in the inferior parietal lobe, you have a distribution which is highly skewed toward contralateral space, so that you can even understand the neural basis of neglect or the motor disorder of neglect. So this is the dorsal reaching system. Because visual input coming from this ear is distributed to area LIP in the inferior parietal lobule and to a number of areas of the superior parietal lobule. This is the end dominant domain of the superior parietal lobule, which project to the both the cingulate domain and to the output motor domain of the frontal cortex. And you have similar things for the other processing stream. But what this picture shows, which is going to be the topic of my talk, is that many of these domains or many of these areas, not just motor cortex, have a direct access to the spinal cord. And in addition, they project to, at least for the control of the eye movement, to the superior colliculus and mesencephalic reticular formation, where Klaus Peter found a lot of reaching-related neurons, which then project for the eye to the brainstem oculomotor center and for the limb to the spinal cord. Now, if this is somehow the structure of the network, what about timing? which in principle is the basis of any dynamic interaction between these areas. Timing is a delicate issue. Some years ago, with Giorgio Innocenti, we started a systematic analysis of cortico-cortical connectivity, starting from interhemispheric connectivity and then also analyzing intrahemispheric connectivity, where using anterograde tracer like the one that Katie has just shown you, we characterize the axon diameters of callosal connection, which are in blue, but also some intrahemispheric association connection. These are the connection that from area PC at the parieto-occipital junction goes to M1, F2, and the cingulate motor areas. And you see that these projections are characterized by axons within a wide range of diameters, which means that you also have wide range of conduction velocities. And of course, what we see here is that, of course, those addressed to motor cortex are thicker. They are very similar to those, but there is an addition of a small contingent of big axons. And this, of course, pose the question of how these things are reflected at network level. Now I'll move to the issue of processing speed. First of all, in the visual pathway that you all know very well. And then within the parieto frontal system, and you see this is neural latency to the visual stimulation in LGN, V1, and the areas of the ventral stream, some prefrontal areas, frontal eye field, and you see that as you move over time, you see a systematic shift of the latency, but you also see rather early response of MST, which in principle should not be somehow expected, while the other areas are come later. Very little is given. Very little information is given for the parietal cortex here, which, while we can get some more information probably by studies like these are local field potential recorded in several different regions of the brain, while the monkey was performing a gonocole task in which he had to discriminate two different objects, shape of different object. And the results were that if you compare the onset time of the change in the local field potential relative to those of visual cortex, you might get surprisingly early response in motor cortex, okay? And you might get dorsal premotor cortex coming later than motor cortex, which is unexpected, and the frontal eye field, at least the lateral frontal eye field, stays in between. Prefrontal cortex is very late. In this study, I want to just refer you about the inferior and superior parietal lobe. This is motor cortex, and you see that the inferior parietal cortex is on average activated almost at the same time of striate cortex, and the superior parietal in this case is a little late. And in this study, the conclusion was that the earlier response to stimuli appeared throughout the cortex after about 50 milliseconds, and the activity began to predict the monkey choice after approximately 150 milliseconds in a distributed network that include both parietal and frontal areas. Now, this is an experiment done by Alexandra and Philippe Archambault in my lab some years ago, where they trained monkey to reach the visual target, and during reaction and movement time, the target was shifted in place, and the animal had to adjust the movement trajectory, and they have been recording in dorsal premotor, motor, and parietal cortex. And what you see here is that this is the neural activity during a single-step trial when the monkey reached to a visual target and conclude his movement. This is neural activity when the targets shift in place, and this is the moment when they diverge in time. Okay, so there is a bump of activity when the new target call for a reupdate of the hand movement trajectory. And if you look at the timing, okay, the timing here is that this line here is the moment of divergence of neural activity in the single and double step trials, okay, in premotor, motor, and parietal cortex. And this line here indicate the timing when the end changed trajectory. So of course, neural activity predict in all these areas, but it does it first in premotor cortex, then in motor cortex, and then in posterior parietal cortex. So this means that the onset time of neural activity relative to behavioral event is highly task dependent, and that is very dangerous to make generalization about all these things in terms of timing. Now, I'll skip this image because they say basically the same time, and I want to just give a very short, let's say, interim conclusion about this timing to go to the final part of my talk. So the earliest response to visual stimuli appear at about 30 milliseconds and seem to generate a feed-forward sweep involving extracerebral and temporal areas, which develop a specific stimulus difference within 150 milliseconds, but many motor and premotor and frontal area display activation as early as striate cortex, so this feed-forward sweep through the entire cortical visual hierarchy is not an obligatory way for the activation of frontal cortex, since input can go through pulvinar or lateralis posterior, depending on whether superior or inferior parietal cortex. And so the temporal relationships are highly dependent, and this also implies that to reach the peripheral motor output, the passage through primary motor cortex is not an obligatory step. And that command mechanism can be based on discrete local network within the parieto-frontal system. Now, this can be a rather strong statement that you may avoid motor cortex to go to the spinal cord or to generate command function, but the image of all these processing streams that I've showed to you is a very crude representation of what these things are. You cannot imagine this processing stream as obligatory pathways that have to be crossed whenever we have to make a reaching movement or whenever we have to perform a certain task. This pathway can be used for many different purposes. And one of these is one that, again, Mickey would probably like it, because it was all started when recording in the VIP, in area VIP, I believe with Jean-René and Carol Colby, they found neurons that have a receptive field on the face, but they were also sensitive to approaching visual stimuli, which were congruent with the location of the receptive field in the face. And this can be imagined as a mechanism by which we detect approaching stimuli, which can be potentially dangerous. I mean, our head is a real estate, I would say, very important, because all the sensory organs that we have stays here, okay, from trigeminus to eyes to ears to olfaction and whatever. So it can be reasonable to believe that you have here a system for danger avoidance. And this system is coherent with the input that VIP takes from PE for somatosensory receptive field, from MST, from vestibular cortex, which can be very useful for defending ourselves during locomotion. And the projection goes to F4. F4 talks to M1, but F4 has his own direct projection to the spinal cord. This projection is not as fast as M1, but we like to think that this projection is useful for presetting the spinal motor apparatus for coming action, for coming fast action. Imagine that when you walk on the mountain, you finally see a snake. So you first stop, and then you have to decide where to go. Should I go or should I stay? And in all these, a snake is a very salient object, I would say. So what you do? You stop and then decide what to do, and then move. And these projections here from V4 to spinal cord, we do believe that it conveys salient related information to the spinal cord. So corticospinal system influenced by saliency of events in our extrapersonal space. And then, you know, this poses a question of recruitment, because these are the corticospinal conduction velocity of F4, M1, as we have seen, and SMA done in a recent study with Giorgio. And you see there is a very big overlap. So that you can imagine that when the fastest corticospinal actions from V4, they overlap with the slowest of M1, so which is recruited. It has to be really decided. It's not taken for granted just because M1 or F4 are activated. You really, the system are really to decide which is within each individual corticospinal system is, well, I would say the task will decide whether you're going to activate the fast or the slow component. And very little information is known about that. This is similar things for the grasping system. Peter Strick have shown a projection from area PEA and from area, and we have shown a projection from area IP to the spinal cord, and this is the conduction velocity. You might have hand action which might not require the direct involvement of motor cortex. Conditional visomotor association and learning. I have learned that since it's red, I should stop. But I'm going to violate, because this is an arbitrary association between the visual input and my action. Okay, so if I am allowed, I can violate, provided it is association that is arbitrary and can be changed. But this is what we do at the traffic light. If it's red, we go. If it's otherwise, we stop. And this is centered on PMD, on dorsal premotor cortex, with this direct access to the spinal cord, and in a distributed system which is involved the striatum, and which also involved the hippocampal formation. Same for internally generated memorized movement sequence from SMA. And finally, well, it's almost obliged to every meeting to talk about the mirror system. The frontal node of the mirror system, RF5, is a direct projection to the spinal cord. It has an interplay with motor cortex. We don't know the conduction velocity of this pathway, but this is the antidromic timing of activation. So these are the median, about 1.5 millisecond or 1.2 millisecond for motor cortex. It's about 2.5 for much more dispersing time. And the idea, this information combined with the fact that the action observation is a suppress half of neurons in area F5 have generated the convincement that when we observe the action of another individual, we inhibit our own action, or when we switch from performance to observation, we switch from one system to another. So now to comply with the red lights, my conclusion is just one, that the multiplicity and heterogeneity of cortical descending system imply that to get to the motor output, we don't need to go every time to motor cortex. And this is also supported by the consequence of lesion, which are different if you have a lesion in M1, in dorsal premotor cortex, in posterior parietal cortex, and so on. Thank you for your attention.
We have a minute. Any question?
I just wanted to stress the superiority of the visual system versus the motor system. Because in the visual system, we have traced a pathways of connections which goes from the occipital cortex in two streams into the temporal, etc. That pathway may be wrong, but for the motor system, you still have even more complicated issues to solve. Does the command originate in the parietal cortex, in the premotor cortex, in M1, and how are all these areas, talking to each other? I think you have to go back and work harder.
No. I think that you have to go back and work harder, because I've been trying to convince you that where the command is initiated depends on the task. So sometimes the command requires the involvement of parietal cortex, some other time requires the involvement of premotor cortex. The shift of the temporal order of activation for reaching to visual target, to simple reaching to visual target, when you change target location so that the entire motor program has to be reupdated, is a very clear example. So what has to be done, Giorgio, really, I believe, is in front of this huge heterogeneity of function, we need network analysis, of course, but also we need to stress the system using multiple tasks. Otherwise, we won't be able to unravel all the richness of the functional properties within this system. This is the only answer, reasonable answer I can give it to you. In my second life, I will go back and look, as you say.
So there is a recent paper showing that another reaching of visual target is actually reaching by your eye gaze. Yeah, you don't necessarily reach by your hand. Something appears in a visual field, you look at it. That's a reaching, isn't it? Reaching by looking at it. And recently there's a paper showing that in monkeys, this signal comes from V1. This is the first visual cortical area, anywhere, whether it's visual or motor, whatever, to make a saccade to it. Yeah, although it's a long latency, it's 40 milliseconds when V1 already shows that signal where it reaches to. That means if you saccade to it faster, the V1 signal is higher. When you saccade to it slower, V1 signal is slower, even though the visual input on the screen is exactly the same. So those are repeated trials over interview with other trials. Monkey didn't know, of course, they're the same input. It could be in between two same input as other visual inputs. But if in one trial the monkey saccaded to faster, another trial monkey saccaded to slower, the V1 responses is higher and lower accordingly.
Well, these tells that V1 is involved in this process, okay?
Not only is it involved, but also it's so fast. It's 40 milliseconds. You cannot have this.
But you know that there are...
Yeah. Because it's too late by the time you look at it. 40 milliseconds after visual onset.
Yeah,
and monkey doesn't know where the target will be, and they just saccade to it.
Yeah.
So where could it be, the signal coming from? Except for V1 actually is creating it and generating
I
frontal
don't know.
retina?
You know, there are ultra-fast responses to visual stimuli also in the frontal eye fields, which has been shown by Simon Thorpe, I believe.
Before 40 milliseconds in V1 the signal? It's
well,
40 milliseconds in V1,
I don't
so
know.
where could it be?
I'm not claiming that V1 is not involved. V1 is not always, might not be always involved. You know, it really depends on the task.
In this case, it's before any other area. Just.
Last question. Just talk.
Okay, just a quick comment and a question. I think that all neuroanatomists would like to go back to the lab and work harder, but there is a need for more funding for neuroanatomy, especially in monkeys. One question I had for you about the clusters that you have. All the areas that part of a cluster seem to be organized based on neighborhood relationships. So it's really rare that they are distant from one another. Yet in previous studies, for example, from Joel Price, he would show that in this orbital network, for example, there would be some islands belonging to the medial network. Do you think that this type of heterogeneity exists, or is it just an anecdote? And what sense does it make?
No, I believe that this heterogeneity exists. And the orbitofrontal one is an example, and the different colors, which might not have been really appreciated, of the orbitofrontal cluster says that within the same clusters there are areas which are those which do not belong to the real let's say, orbital part of prefrontal cortex, but stays on at the edge between the orbitofrontal cortex and the lateral part of prefrontal cortex, which stays together, which clusters together, and which seems to have a different function relative to the other one. This is what is supported by this connectivity. And then, yes, it is true that these clusters very often put together regions which are topographically close, one relative to the other, but which do not respect architectonic boundaries somehow.
So you think there is no relationship between the connections of these regions and the architectonic delineation?
No, sometimes you can recognize of individual areas. Nobody has ever attempted to see whether the areas that belong to a cluster, they have common architectonic or cytochemical features, okay? So if this is your question, I cannot answer you. For sure, there are parietal areas that you can recognize fairly well based on architectonics. And in this case, you can add additional information based on cortical connectivity.
Okay, thank you.
So we have to end the session. Thank you very much again to the speakers and to all those who discussed.